So why microbial community editing is important? Because these microbes are affecting the host they're living in, and understanding how these microbes are contributing to health and disease will help us understand more what they're doing in the human health and also in the environment. And the top example is one of my favorite examples because microbes living in the rumen gut are one of the major producers of methane. And understanding and controlling these microbes could get us a step closer in controlling and sort of mitigating climate change related problems. So the proof of principle that our lab has developed involves community editing of the microbes in a small community. So the very first step is targeting a non-targeted or randomly inserted vector, just to see which microbes in the community can be edited, which are genetically tractable. And we call this method as ETC, shown here as 1. And once we've established which microbes are editable, the next step is to actually edit them or target them. So for this, we use a targeted vector so that we can insert the gene of interest in these microbes that are tractable. This method we call as DART. Now that we've established that this method works, the next step is to improve it, right? We can still do it in a very small community. We can target only a handful of microbes. We want to improve it. We want to make sure that we can go in and look at any hot spring environment and sort of understand what these microbes are doing. Very briefly, before I start talking about what my part in this project is, I want to introduce a concept because I'll be using that a lot. The targeted vector, and the D here is somehow hidden, DART that I just talked about is actually a CRISPR-Cas transposon. Everyone might have heard about CRISPR-Cas and how it's used for editing. But CRISPR-Cas transposon is like the next step of editing because it allows to precisely edit the system without using the host for repair. So it has a much cleaner and less messy editing than CRISPR-Cas. And I'll be using this a lot because we've used this CRISPR-Cas transposon system as a targeted vector for our community editing. So just bringing it all together, we have, as a lab, developed a proof of principle for community editing, but we want to improve it. We want to see if we can target more non-model organisms and improve the efficiency of the organism that can be targeted. So in order to do so, we thought that, okay, what if in the targeted vector that we're inserting, we add a regulator, we add a gene that can help improve the efficiency of this vector. We call this a regulator. And after adding this regulator, this would help target more of the microbes of interest and do a better community editing. So our lab has done some whole genome screens in E. coli to find these potential regulators, which after being targeted by DART, either increase in their relative abundance, which we call as activators, or decrease in their abundance, or which we call as inhibitors. I say we, but this project was led by Leo Song and the amazing scientists and researchers at Berkeley and Rubin and Crest Lab, and we also have one, Amanda, sitting here. So these regulators or activators and inhibitors that the lab has found is what I'm using. And my step in my work in this project is we have done whole genome screens in E. coli, found these regulators. How conserved are they? Can we use the presence-absence profile of these regulators in non-model organisms or non-model strains to see this gene is more present, so maybe this is a regulator in non-model strains and we can target that. Or we're like, you know what, this is E. coli specific, it's missing, so we don't care about it. So sort of bioinformatically see how conserved these regulators are and then use that information to expand the host range of targeting. This is the very first analysis that I did. So I went into the literature, found 11 genomes for which there is experimental evidence that the DART vector works or not work, shown here in black and gray. On the x-axis, I'm showing the presence-absence profile of the regulators that candidate regulators that we found, which can be subclassified as activators and inhibitors. And just the very first pass in these 11 genomes for which we have the experimental data, we find 5 different genes that are present in all the 5 genomes. So they're sort of conserved. And 4 different genes which are present mainly in the group in which the DART works. These are the genes that we want to look at because we want to know which candidate regulators are important for DART insertion. So 11 genomes, a very small data set, but we have found some conservation profiles. Some genes that are group specific, some genes are present in all 5 genomes. So the next step was, what if we look at a larger scale? Can we find similar trends if we look at a larger group of data set? And can we then use this information to find some non-model strains to edit? So because there's not much information on experimental data where DART works, we use in bioinformatic data as a proxy. If the genome has the DART genetic architecture, we classify them as DART containing. And if they don't have it, we classify them as non-DART containing on the left. So again, this helps improve the data set that we have, but we have to take with a grain of salt that this is a bioinformatic data, and we're using the information that DART works or not based on the presence of the genes for DART. And with the 2 data sets that were publicly available, we now have about 2,500 genomes to examine. I examined the presence-absence profile of these same regulator genes shown here on the y-axis this time, subclassified as activators and inhibitors in blue and red, and just looked at the number of genomes in which these regulator genes are present. And interestingly, some of the profiles were conserved. The 5 genes that were present in all the 5 strains, 11 strains that I looked at before, are also present in more than 99% of the samples here. So we can safely say that these genes are conserved. They are important for cellular processes, which they are. IHF-A and IHF-B, for example, here in the middle, are DNA-modifying enzymes required for recombination. You need them regardless. At the same time, the other 5 genes that we found were group-specific in the experimental data, 2 out of those 5 genes actually show a significant variation in these groups, the DART and non-DART groups, YDHO and YDIP. And these 2 are now our targets or candidates to verify and sort of see, are they even doing something or not? So sort of conclusion from this data set is that we found, very interestingly, the presence-absence profile of the regulator genes to be conserved in experimental and bioinformatic data. And at the same time, found some candidate genes that we can then experimentally validate as important or not for DART insertion. And the 702 genomes that classified as DART genomes could now be our potential non-model candidates that we can now go in and see if we add these regulators to the increase, can we increase the targeting efficiency or not? So in conclusion, our lab has identified through whole genome screen some candidate regulators, and then we went in bioinformatically to find out of these regulators which can be targeted and which host strains, novel host strains we can examine. And this fits in beautifully into the big major goal that we have in the lab to improve microbial community editing. And this I'd like to thank our lab, Rubin Lab at IGI, and our collaborators at IGI and externally for their support and advice. And with this, I'll take questions.
Okay, any questions from the audience? Do we have any online questions? Okay, for the sake of time.
Thank you.
Thank you. Oh yeah, let's thank Gina.